Hello students, it's 3 p.m. so the class would commence soon. Today we'll be dealing with lecture week 3 and problem set assignment. Let's wait for 5 more minutes and start the class presentation at 3:05. Meanwhile, I'll keep admitting students who are requesting to join this call. Hello students, hello again. So it's 3:05 now. We will begin with the presentations. So I'll just share my screen with all of you guys. And meanwhile, we have a thin population. If more students want to join this call, they are free to do so. I'll keep admitting anyone who requests to join this call. Meanwhile, let me share my screen with you. Let me open my presentation. Yep. So, hi everyone, and welcome to week 3 for Basics of Biology, NOC 25 BT02. In week 3, we will solve all the questions related to biotechnology techniques and cell cycle and electron transport chain, the chlorophyll molecule, and the instructor for this course is Professor Trivedi from IIT Guwahati, and my name is Ashutosh. I'm a PMRF scholar, and I am a PhD student at IIT Kanpur. So some basic set of instructions for all the students as well as those who will be watching this video. This is a recorded class. The presentations will be uploaded in a drive link which will be shared with you via the NPTEL team. And through this class, we will solve 10 to 15 exemplary questions from all the lectures you have encountered in week 3. Now, if you're following with the Swayam NPTEL course, then there are multiple lectures covered in week 3. And this problem solving session will give you a taste and also act as a recap for all the topics that you have covered in the lecture week 3. And this problem set solving assignment will help you solve your real assignments when you are submitting your quizzes and assignments for the course for grading. Yeah, for all those who have joined new, I would like to go through all this again. So in this will be an interactive session, but the interaction with me will happen through the comments or the chat box. I would request all the students to turn off their video and mic during the presentation for smooth conduction of this class. But don't you worry, this will be an interactive session. You can interact with me via the chat box. So we'll be solving 10 to 15 exemplar questions, which are not the real assignment questions, but related to the topics that you are going to face in the assignment or in the week module, right? This is a very simple and easy to understand course, Basics of Biology, and I'm here to help you out in case you have any doubts. And meanwhile, when we solve these questions, we will also cover a brief about those topics of which these questions are designed. So okay, as we follow through this session, long session of 2 hours, we will see, and it will be fun. Cool. So meanwhile, we are 5 to 6 students just now. Let us hope we get more students on the board. If not, these recordings will be available in YouTube. And at the end of this class, upon request, I'll share the YouTube link with you. You will get provided with all the recorded assignment sessions from the previous classes as well as upcoming weeks from the YouTube playlist channel. And these slides will be shared with you for reference through the NPTEL team. Okay, with that much of introduction, let's begin with the first question of this session. First question is displayed on the screen. When an isolated plasmid is run on an agarose gel, what is the order of its appearance? Understand the question carefully. When an isolated plasmid is run on an agarose gel, what is the order of its appearance? Option A, loading well, covalently closed circular DNA, open circle, RNA, supercoiled DNA. This is option A. Option B, loading well, covalently closed circular DNA, open circle, supercoiled DNA, RNA. Option C, loading well, open circle, covalently closed circular DNA, RNA, supercoiled DNA. And option D, loading well, open circle, covalently closed circular DNA, supercoiled DNA, RNA. So here I have presented with a question and 4 options. I would request all the students to write in the chat box which they think is the correct answer or correct option for this question. You can just mention option A, B, C, D. Go through the question, let's wait for some time and then collect some answers. And after a minute or so, I'll reveal the correct answer and we'll also understand the explanation behind the correct answer. So to denote loading well, these arrows denote the position of the position of the products from top to bottom, right? The right arrow is, these arrows are on the top, well these products are on the bottom. From top to bottom, what is the arrangement when you run an isolated plasmid on an agarose gel? So all these topics are from the same lecture week 3. So let me wait for some time and I want you guys to understand this question. Read all the options carefully because options can be confusing sometimes. And I'll keep an eye on the chat box to collect your answers. You can just mention option A, B, C, D, whichever you think is the correct option in the chat box. No answers yet? If the question is difficult and you do not know the answer, but you can just guess, you can take a wild guess based on logic, that where do you think all these products would appear in the gel when you run an isolated plasmid? Okay, I see one answer. Vedashri says option A. Okay, let's not lose any more time and time to reveal what is the correct answer for this question. The correct answer for this question is option B. So when an isolated plasmid is run on an agarose gel, this is the order. From top to bottom, the products will appear. First will be the loading well, then covalently closed circular DNA, then an open circle or a nicked circle, then supercoiled DNA, and then RNA. Right? So this is the correct order of products in the gel. Now what is agarose gel electrophoresis? Agarose gel electrophoresis depends on the principle that larger sizes of nucleic acid or DNA or large molecules travel slower through a agarose gel. Now agarose is a substance which is solid at room temperature but liquid at higher temperature. When you melt agarose can be a gel like a substance. And the principle is agarose creates a porous membrane or a porous surface through which DNA and RNA can have a sieving effect. It can move through the porous gel. Now, which bands will move faster and which bands will move slower? So, larger bands will always move slower and smaller bands would move faster. And not just this, any modification or any form which hinders the speed inside an agarose gel or offers more resistance to the molecule will have a slower movement. So agarose gel electrophoresis is a molecular biology technique to separate DNA or RNA fragments based on their size. So it is a size based distinction method where you can distinguish different lengths of DNA and RNA. It utilizes an electric field to move negatively charged nucleic acid through an agarose mix. So this is its principle. When you have electric field, the electric field is created by positive and negative charges. So the positive electrode will attract the negatively charged nucleic acids like DNA and RNA. Because of the phosphate backbone, which is negative, nucleic acids are generally negatively charged. So the positive electrode will attract the DNA or RNA, while the negative electrode will repel the DNA or RNA. So this is the procedure by which an agarose gel is made, or a agarose gel electrophoresis. We have a casting tray here with a glass plate, and you use these, seal these ends with a tape and pour the heated agarose solution. And depending on the percentage of agarose gel you have, you can resolve different sizes of band. A higher percentage gel, a higher percentage of agarose gel will resolve very low molecular sizes band, while a lower percentage of gel will not resolve as much. And in a higher percentage of gel, there is much higher resistance for the DNA or RNA molecule to move. That is why higher percentage of gels are slower. Now when the agarose has solidified, before the agarose has solidified, you apply combs. Now these combs will create wells inside the agarose gel into which the DNA can be loaded. And when an electric field is applied as such that the negative electrode will repel the negatively charged DNA or RNA, so the DNA will move away from the negative electrode while it is attracted towards the anode, which is the positive charge, so it will move towards the anode. And then in meanwhile, a mixture of DNA can be resolved depending on the molecular size or the base pairs. Number of base pairs the DNA has determines the molecular size. So after loading into the wells, we run it on a electric field or inside a current, and then the DNA or RNA can resolve itself. Now the principle is DNA and RNA are negatively charged due to their phosphate backbone. When an electric current is applied, nucleic acid molecules migrate towards the positive electrode. Smaller fragments move faster through the gel, while larger fragments move slower, allowing size-based separation. So this is the basic principle. Now what will happen when we run a plasmid in a gel? So this is how a plasmid in a gel would look like. So on the left side, lane M is the marker or the ladder. Then we have 4 lanes. This is the plasmid. So here we have the nicked circular followed by the linear plasmid and the supercoiled plasmid. So that was the option. If you remember the option, the first lane was the loading wells, then was the open circular DNA. What was the option? Covalently closed circular DNA. Yes. So this is the covalently closed circular DNA, like when the plasmid is a circle. So when the plasmid is circle, it will appear somewhere here in the gel because it is a bigger gel. It is of bigger size. Then we have open circle, supercoiled DNA and RNA, right? So open circle, when you have an open circle, it means that there is a nick or a cut inside the plasmid. Then that would appear, that would face more resistance. So nicked circular is appearing here. Then we have linear or double stranded cut. Now when the plasmid is cut and linearized, now it faces less resistance. So it is moving faster than the nicked or circular DNA. Then we have supercoiled DNA. Supercoiled DNA has larger degrees of freedom. That is how it is moving fastest. So it can move through the pores in the agarose faster and therefore appear as the lowest band. And then comes the RNA, which is smaller in size and molecular weight than DNA. That is why RNA will come at the end. So from the top we have the loading wells, then the full plasmid, then the nicked plasmid, the linear plasmid and the supercoiled plasmid. So supercoiled DNA is the fastest. It is the most common form in vivo. So when a plasmid, as we see in the map, it does not exist like that. It exists in a supercoiled state in most forms in vivo. It is a compact structure, moves fastest through the gel. Nicked DNA, single strand break in one of the DNA strands, loosely coiled, moves slower than the supercoiled. And linearized occurs when the plasmid is cut by a restriction enzyme, moves between the supercoiled and the open circular forms. So that is why we have our correct option. First the closed plasmid, then the open plasmid, then the linear plasmid, the supercoiled plasmid, and then comes RNA. I hope it's clear to everyone. If yes, then just give me a thumbs up or let me know in the chat. Then we can move to the next question. Great. Great. Okay. Moving on to the second question. Where does transcription take place in an animal cell? Where does transcription take place in an animal cell? Option A, nucleolus. Option B, ribosomes. Option C, nucleus. Option D, Golgi apparatus. Now these are different cell organelles in an animal cell. You have to tell me where does transcription takes place in an animal cell. Is it option A, nucleolus, option B, ribosomes, option C, nucleus, or option D, Golgi apparatus? I'll wait for some time before looking at the chat box. So since these are recorded lectures as well, or recorded assignment sessions as well, for people who will be looking at them in some other amount of time or in point in the future, they also need some time to solve these questions. So this is the format that I'll keep following throughout these sessions. Where does transcription take place in an animal cell? Let's check the answers. Okay, seems like everyone has answered. Vedashri says C. Aishwarya is saying nucleus. Mrs. Babu and Farsana are also locking C. Okay. So let me tell you that you guys are absolutely correct. The correct answer is option C, nucleus. Nucleus is the place where transcription takes place in an animal cell. So okay, here what I have is a zoomed in image of a nucleus or an animal cell. The animal cell has the outermost membrane, which is known as the plasma membrane, made up of lipid bilayer. And then another very specified organelle inside the animal cell is called nucleus, which is bound by nuclear membrane made up of laminins. Now if you remember the process of transcription is generation coding of DNA into RNA, generation of a messenger RNA from DNA, which we call transcription of genes. So in the coding region of DNA, transcription happens, and all this transcription, since the DNA is getting coded into mRNA, now the DNA is present as chromatin inside the nucleus. That is why transcription also happens inside the nucleus with the help of these special proteins called polymerases. Now this is a DNA dependent RNA polymerase. DNA dependent since it depends on DNA to copy the information into RNA. So it is a RNA polymerase. Now the RNA polymerase, the DNA, the chromatin, everything is present inside the nucleus. Now after generating a messenger RNA, and after the messenger RNA is processed, that messenger RNA is moved out of the nucleus through these nucleopores, and now the process of translation can start in the cytoplasm, and translation is done by ribosomes. So the correct answer therefore is transcription happen in nucleus, whereas translation happens in the cytoplasm on ribosomes. So here is a 3D cross section of a cell showing different organelles, all the organelles that we have encountered in the option. So nucleolus is another structure which is present inside the nucleus. What is its function? Main function is to make rRNA and ribosome subunits. So we have different kinds of RNA in our cells, in any animal cell. We have the main kind of RNA, which is messenger RNA or mRNA. Then we also have non-functional, sorry, non-coding RNAs like rRNA, tRNA, shRNA, snRNA. So rRNA is nothing but ribosomal RNA. So this RNA is a constituent of the ribosome. So nucleolus has the function in making rRNA or constituent of ribosomes. The Golgi apparatus is present inside the cytoplasm, and it is a Golgi apparatus is a sorting factory of the cell. It sorts different proteins. It's a warehouse. It will tell which protein to go where. It modifies, sorts, and packages proteins. Nucleus is the center of the cell. It stores DNA and controls gene expression, which was the correct answer. Ribosome is on the cytoplasm or the rough endoplasmic reticulum. It is on the two surfaces and it is functioning in making proteins. It is the protein synthesis center of the cell. So uh, these are all the four organelles that we encountered in the option, and the correct answer is transcription happens inside nucleus. Is it clear to everyone? Just let me know in the chat. Great. I hope so. This is the fashion in which I conduct classes. I hope with every, even the simplest of the questions, we brush up many other topics that that is covered in the lecture series, so that your memory gets refreshed and you get help while solving your own assignments. Okay, moving on to the third question. Which portion of the mitochondria contains the enzymes required to run Krebs cycle? Now, this is a question. Which portion of the mitochondria contains the enzymes required to run Krebs cycle? Option A, cristae. Option B, intermembrane space. Option C, inner membrane. Or option D, matrix. Now, this is a slight memory based question. If you guys remember Krebs cycle, then you will be able to answer this. Which portion of mitochondria? So, all these options are parts of mitochondria. Which portion has the enzymes required to run Krebs cycle? Indirectly, it is hinting you at which compartment of the mitochondria hosts Krebs cycle, or where does Krebs cycle happen inside mitochondria. Option A, cristae. Option B, intermembrane space. Option C, inner membrane. Or option D, matrix. Okay, I'll wait for some time and let us see. Check all the answers in the comment box after a minute. I would at least request all of the students not to Google these questions and use your own memory of the class or the lecture or your knowledge. Then it will be more fun and you'll be engaged with the assignment solving session. Which portion of the mitochondria contains enzymes required to run Krebs cycle? Okay, let me check the comment box. Oh, it's great. All of you have answered. Vedashri, Aishwarya, Farsana, and Mrs. Babu are all opting for option D, mitochondrial matrix. Mitochondrial matrix is the correct answer to this question. Well done, congratulations. Mitochondrial matrix has all the enzymes to run Krebs cycle. Krebs cycle, which is also known as TCA cycle or tricarboxylic acid cycle. So let us have a brief overview of the TCA cycle. Why is mitochondrial matrix the main site? Because it includes most of the enzymes like citrate synthase, aconitase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, succinyl CoA synthetase, fumarase, and malate dehydrogenase. Right? So this is the overview of a Krebs cycle. So in the Krebs cycle, we have our tricarboxylic acid citrate, which is then treated on, tricarboxylic acid, oxaloacetate, then we have citrate synthase turning into citrate, aconitase to isocitrate, just rearrangement inside the citrate. Then we have isocitrate being changed into alpha ketoglutarate. Then with the help of the enzyme isocitrate dehydrogenase, and here we have alpha ketoglutarate dehydrogenase turning alpha ketoglutarate to succinyl coenzyme A, and then succinyl coenzyme A is turned into succinate here with the help of utilizing an ATP. Succinate then turns into fumarate, and fumarate to malate with fumarase, and malate to oxaloacetate via malate dehydrogenase. So all these enzymes, most of these enzymes are present inside the mitochondrial matrix. That is why Krebs cycle happens inside the mitochondrial matrix. Okay. So here is a brief diagram of all the processes happening at the mitochondria. The outer membrane and the inner membrane, and inside is the matrix. So this is the place for the Krebs cycle. And then in the inner membrane are embedded proteins, which are electron transport system proteins, and the ATP synthase, which is the primary ATP producing enzyme, right? So the correct answer is, as all of you have said, the matrix is the site for Krebs cycle. Moving forward to the fourth question, where is chlorophyll localized in the chloroplast? This must be an easy question. Where is chlorophyll localized in the chloroplast? Option A, outer chloroplast membrane. Option B, inner chloroplast membrane. Option C, intermembrane space. Option D, thylakoids. Now, where do you think chlorophyll is localized inside the chloroplast? This should be an easy question. Let us wait for some time and collect answers. Okay, let me take a look at the chat box. Oh, great! All of the students have answered. Vedashri says D. Farsana is saying thylakoids. Mrs. Babu D. And Aishwarya is also going with D. Thylakoids. Absolutely correct answer. Thylakoids is the correct answer to this question. Chlorophyll is localized in the thylakoids inside the chloroplast, which are special organelles present in plant cells but not in animal cells. Chlorophyll is specific or endogenous to plant cells. So here we have a cross section cut open of a chloroplast, which is also called a shoe-shaped organelle. It looks like an oval shoe. Now these are the different parts of a chloroplast. It is a double membranous structure, just like the mitochondria. Then the inside of the inner membrane is stroma, which has carbon fixating enzymes for the light independent reaction or the dark reaction. So the dark reaction happens inside stroma. Then we have disc shaped membranous structure, just looking like buildings inside the horseshoe, which is called thylakoid. This thylakoid, the membrane of the thylakoid, contains chlorophyll for the light dependent reactions. And the stack of this thylakoid is called granum, a stack of thylakoids that increase surface area to volume ratio to optimize light absorption by the chlorophyll. And the connecting links between these different stacks are called lamella. Lamella connects and separates thylakoid stacks, which are also called grana, singular for granum and plural for grana. Okay, so this is the stroma, the thylakoid, the granum, and the lamella. All of these are parts of the specialized organelle called chloroplast, and which harbors the chlorophyll. The membranes of the thylakoid contains chlorophyll. Okay, coming on to the fifth question. Which of the following in the chloroplast is responsible for absorbing photons? Which of the following in the chloroplast is responsible for absorbing photons? Option A, translocon on the outer chloroplast membrane, TOC. Option B, photosystem 1. Option C, translocon on the inner chloroplast membrane, TIC. Or option D, photosystem 2. Which of these is responsible for absorbing photons? Now, if you guys know about chlorophyll, you can answer this question. It's not a difficult question. Let us wait for some time to allow people to give me answers. A minute or so, and then I'll look at the chat box. If you have any questions or any confusion about the answers, or you want to talk about it, you can mention in the chat box. Which of these in the chloroplast is responsible for absorbing photons or light? Okay, let me look at all the answers in the chat box. I think Farsana has answered option B, and while Vedashri is answering option D. Yeah, it's a little bit, might be a little bit confusing between photosystem 1 and 2, but I hope after looking at this explanation, we'll be clear about all our doubts. Now, which of the following in the chloroplast is responsible for absorbing photons? Let us look at the correct answer. The correct answer to this question is option D, photosystem 2. So PS2 or photosystem 2 present in the chloroplast is responsible for absorbing photons, not PS1. And translocon on the outer chloroplast and inner chloroplast are just transporters or channels. Right? Now, what is PS1 and PS2, and how does PS2 act as the first organization to harbor or harness the energy of light? This is the photo system 2, a schematic showing the sun's energy, light energy from two photons first hitting the photosystem 2 antenna complex. Then the photons jump from the antenna complex to reach the what is known as the reaction center. Now the reactions center, meanwhile two other electrons are used on the other side of the photosystem 2 to split water into two protons, right? And now coming back to the reaction center, two electrons two electrons are then donated to the primary acceptor. Electrons are transported. Primary acceptor then transports it to plastoquinone, then the cytochrome complex, and in the cytochrome complex, using the energy of these photons, one molecule, ADP to ATP conversion is catalyzed. So the photosystem 2 is the first protein pigment complex in the light dependent reaction of photosynthesis. So this is the light reaction in the photosynthesis. It is embedded in the thylakoid membrane of the chloroplast, right? So when we were talking about chlorophyll being present in the membranes of the thylakoids, this is where the chlorophyll is present inside the photosystem 2. And this plays a crucial role in capturing light energy and initiating electron transport. So this is the first key event in the electron transport chain inside a chloroplast. Now, what are the series of events? First, the photon is absorbed by the photosystem 2 or PS2. PS2 contains chlorophyll A molecules, mainly P680. So this wavelength is 680. This wavelength of photon can excite this chlorophyll molecule that absorb light energy. When P680 absorbs a photon, it gets excited to a higher energy state. This excited P680 transfers an electron to the primary electron acceptor here, pheophytin. So this primary acceptor is pheophytin, while this P680 is the chlorophyll A molecule present in the photosystem 2. Now looking at the whole electron transport system, or which is popularly known as the Z scheme inside the plants. The Z scheme represents electron transport pathway in the light dependent reactions of the photosynthesis. So this is the ETS or electron transport system in the photosynthesis, light dependent reactions. It shows how photosystem 2 and photosystem 1 work together to produce ATP and NADPH. Adenosine triphosphate, which is the direct energy currency of the cell, and NADPH, nicotinamide adenine dinucleotide phosphate, which is also a currency which can be used later used as an energy, which are needed for the Calvin cycle. So this feeds into the Calvin cycle or the Krebs cycle. The name Z scheme comes from the zigzag shape of the electron energy levels in this process. So first the reaction starts when light energy hits P680, the electron is excited. Electron is then passed on to the first carrier, which is the pheophytin. Pheophytin then transports it to the plastoquinone, and plastoquinone gives it to the cytochrome B6 complex, cytochrome B complex. Cytochrome B complex then transports it to the photosystem 1. So this here is photosystem 1, which is P700. When the electron is now passed here, P700 gets excited, then it transports its electron to phyloquinone, then iron sulfur protein clusters, ferredoxins, flavoprotein, thereby creating an NADPH molecule which can now be used for fueling the Calvin cycle. So this whole scheme of electrons transporting from PS2 to PS1 using the light energy is known as light dependent reaction in the photosynthesis, or popularly known as the Z scheme. So the answer to our question was light absorption first happens at photosystem 2 during the light dependent reactions. Right? Is that clear to everyone? Just let me know in the chat and we will move to the next question. Very good. Very good. So this is a common confusion that students do have. But once you get thorough with the electron transport system and the light reactions and the Z scheme, you will be able to differentiate which is the protein system which absorbs light first. Cool. So far so good. Moving on to the next question. Question number 6. Which pH indicator is used in DMEM media? Okay, this is an interesting question. Which pH indicator is used in DMEM media? Option A, methyl red. Option B, phenol red. Option C, neutral red. And option D, thymol blue. Which of these indicators are used in DMEM media? Now I'm not sure whether you have seen what a DMEM media looks like and what is it used for, but if you have, then this answer is a piece of cake for you. Let's wait for some time and collect some answers. After a minute or so, I'll take a look at the chat box. Now, which is the indicator that is used in DMEM media? Okay, let's take a look at the answers. I'm sorry. Vedashri is guessing B. And what about others? Mrs. Babu has answered B. Okay, let me not take much time and reveal what is the correct answer. You guys are both right. It's option B, phenol red. So, phenol red is a pH indicator that is used in DMEM media. Now, who can tell me what is DMEM media? I'm sorry. Can you post it in the comment if you know the full form of DMEM media? If you do not, then that's fine. Just let me know. I just have the expanded form in the next slide as well. Do you guys know what is DMEM media? Where is it used for, or at least the full form of DMEM media? You can mention in the chat box. Yeah, Mrs. Babu has tried her best. She doesn't know the first two expansions, but Eagle Media. Oh, great. We have a correct answer. Vedashri is correct. Yeah, it's fine. It's fine. Vedashri has the correct answer. Dalbeco modified Eagle medium. Yes, so DMEM is also known as Dalbeco modified Eagle medium. DMEM. Now, what does this media is used for? Dalbeco is a person who has identified or discovered it. Sorry, Dalbeco is the person who modified Eagle's medium. Now, Eagle is another scientist who discovered this media, and then Dalbeco added his modifications to make this complete media for cells. So, this media is used repeatedly and regularly for in vitro cell culture. So, when you culture cells on a plate or a petri dish, you need this media. This media has the indicator phenol red, which looks like this. Phenol red can change color. So all these indicators change color when there is change in pH or basicity or acidity, right? So phenol red is yellow at low pH or acidic pH when the pH is less than 6.5, while it is red or orange red at neutral pH 7.4, which is also the pH of blood or the cellular milieu, optimal for most in vitro cell cultures. And when it is highly alkaline, it is pink or purple, dark pink or purple. This helps researchers monitor the pH of the culture medium. Now, why do we use this Dalbeco modified Eagle media with phenol red? Now, it can tell us when to change, either to change today or check for contamination in our in vitro cell culture, or you can change it within 1 or 2 days, leave it because it is at the correct pH at 7.4, or change or regas the CO2 or check incubator CO2 supply when there is highly alkaline medium. So this indicator can tell us immediately whether the cells need immediate changing, or there is a decrease in the CO2 supply in vitro cell culture. That is why we used DMEM with phenol red. So all the packs of DMEM that you buy or any pre-made media that you buy is always red to pink in color, which is the color of phenol red. Good. You guys have answered correctly. Moving on to the next question. Question number 7. The interphase of cell cycle comprises of dash. Now this can be a tricky question as well. The interphase of cell cycle comprises of option A G1 S G2, option B S G2M, option C G2M G1, and option D G1 G2M. Now which of these is comprised by the interphase in the cell cycle? Let's wait for some time to collect some answers. I saw new people who had joined this call. So, for people who have joined this call recently, as well as other students, I would re urge all of you guys to participate in this discussion. While I keep posting the questions on the screen as well as I verbally announce it for you, you please let me know what do you think is the correct answer for this question in the chat box. You can just mention your option, or if the words are less or the answer is short, then you can type it in the chat box. I will follow up with the chat box in a minute or so. The question is, which cell cycle phases comprise the interphase in the cell cycle? Okay, let me look at the answers in the chat box. Aishwarya, Farsana, Mrs. Babu, Vedshri have all answered option A, G1, S, and G2. That's great because this is the correct answer to this question. Well done. Very good. This is the correct answer to this question. Interphase is comprised by these three phases in the cell cycle, which is G1 phase, the S phase, and the G2 phase. Now, these are the total phases inside a complete cell cycle. We have the cell cycle can be broadly divided into two phases, the interphase and the mitosis phase, right? Now, the interphase is the phase where the cell grows and copies its DNA. Now, before actual division of the cell, which is the mitosis, mitosis is the where the cell divides in its DNA and cytoplasm forming two new cells. Now, before forming actual two new cells from a single cell, the cell needs to replicate its DNA. The copy of DNA has to be made, new copy of DNA has to be made. It has to synthesize a lot of proteins and the volume of cytoplasm before even actually dividing, right? So, all these activities before the actual division of the cell is coming under the interphase. So, the interphase can then be divided into three more phases: the G1 phase, where there is cell growth, S phase, where there is DNA synthesis, and G2 phase, which prepares more growth and preparation for mitosis, right? And then similarly, mitosis can be divided into five more phases: prophase, prometaphase, metaphase, anaphase, and telophase. And there is another special stage, the G0 resting stage, where the cell performs its function and is not preparing to divide. So these are all the stages of the cell cycle, and the interphase has all these three phases: G1, S, and G2. G1 stands nothing but growth 1 phase, S is the synthesis phase, and G2 is growth 2 phase. Is it clear to everyone? Let me know in the chat box. I think this is the explanation I have. Yes, very good. Question number 8, the next question. The stage of mitosis where nucleolus disappears is dashed. Now, which is the stage of mitosis where the nucleolus disappears? Option A, anaphase. Option B, metaphase. Option C, telophase. Option D, prophase. Now, in which of these phases of mitosis do you think the nucleolus disappears? I will wait for a minute and then look at all the answers that you have posted. Please take your time and think about the right answer. Post it in the chat box. I would encourage maximum participation. Please do not shy away from these assignment and live sessions, because this is the only point of live session you have with any kind of NPTEL courses, right? So please take the most advantage out of it. Participate in the discussions. Let me know what you think is the correct answer, and we will move forward. Okay, I'll wait for a minute and then look at the answers. Okay, let me look what are the answers that have been posted in the chat box. Mrs. Babu says D. Aishwarya is saying D. Farsana is also saying prophase. Vedashri says B. Metaphase. And Rishikesh has answered A. Anaphase. Okay, let me tell you what is the correct answer to this question. Correct answer is D. Prophase. Very good. Congratulations to all those who have answered correct. Prophase is the stage where the nucleolus disappears in the mitosis. Okay, so what are all the stages of mitosis and what are the characteristics of these phases? Let me emphasize stage by stage for the full cycle of mitosis. Now, the first phase is the prophase, right? Now, what are the characteristics of prophase? Chromosomes condense and become visible. So, when you observe a cell in prophase, you can visualize the chromosomes. They are condensed, they are compact, and they have become visible. Spindle fibers emerge from the centrosomes. Now, the centrosomes are microtubule emanating proteins. Centrosomes are organelles from which spindle fibers will originate and attach to the chromosomes, right? That is how the chromosomes will be pulled apart and separated into two different cells. Now, the nuclear envelope breaks down. So, if you remember the earlier questions where we talked about, the cell has an envelope called the plasma membrane, while inside the cell, in the nucleus, that also has a membrane called nuclear membrane or the nuclear envelope made up of proteins, while the plasma membrane is made up of lipids, and the nucleolus disappears. So you cannot see the nucleolus. So this is the correct answer for the question. Prophase. Then what happens in pro-metaphase? So pro-metaphase is a transition between prophase to metaphase. Many literature does not consider this as a proper distinct phase in the mitosis. But what happens in pro-metaphase? Chromosomes continue to condense. Now they thicken much more. Kinetochores appear at the centromeres. Now the centromere is the point in which the chromosomes are jointed. The sister chromatids, 4 chromatids are jointed. So these areas are called centromeres. Now in centromeres there are regions called kinetochores consisting of high protein density which appear. And mitotic spindle microtubules attach to kinetochores. So the spindle fibers which are originating from the centrosomes will now attach to the kinetochores or to the center of these chromosomes. Centrosomes move toward opposite poles. They kind of slightly orient themselves towards the opposite pole. Now comes the metaphase. Mitotic spindle is fully developed. All the fibers, the nucleus is totally gone. Centrosomes are at opposite poles of the cell. Chromosomes are lined up on the metaphase plate. So the metaphase plate is which is very characteristic to a cell cycle metaphase. Each sister chromatid is attached to a spindle fiber originating from the opposite poles. So a single spindle fiber attaches to only one kinetochore on one side of the kinetochore in a chromosome, and from the opposite pole there is another spindle fiber which is attaching to the other side of the chromosomes, so that they do not attach to single side rather than two opposite sides in a chromosome, right? Then comes the anaphase. Cohesin proteins binding the sister chromatins together break down. sister chromatids are pulled towards opposite poles. So the proteins which are binding the chromosomes, which are creeping the chromatids, all the four leaves of the chromosome together, which are called cohesin proteins. These proteins break down and which allows further separation of the chromosomes. So anaphase is a stage where you can visually see the chromosomes moving towards two opposite poles, right? And then comes telophase. Telophase is reappearance or reformation of the nuclear membrane around two individual groups of chromosome inside a singular cytoplasm. So this is a state in which temporarily in a single cell there exist two different nuclei. Chromosomes arrive at opposite poles and begin to decondense. Nuclear envelope material surrounds each set of chromosomes. The mitotic spindle breaks down. Now the mitotic spindle is slowly breaking down. So this is the completion of the what is known as karyokinesis. Karyokinesis means successful nuclear division. And then which is followed by cytokinesis, which is the actual cytoplasmic division. The separation of cytoplasm into two different cells and the rupturing and puncturing of the plasma membrane and rejoining of the two cells. In animal cells, there is a cleavage furrow which separates the daughter cells, and in plant cells, a cell plate separates the daughter cells. So these are all the different phases and their characteristics in a single mitotic cycle. And the answer was prophase, which is where all the nuclear structures disappear, like the nuclear membrane as well as the nucleolus. Is it clear to everyone? Since there was a little confusion about all the answers, I found out. Good. Great. Great response. Let's move on to the next question, which is a related question to the cell cycle. Which of the following is a characteristic of metaphase? Which of the following is a characteristic of metaphase? Option A, the chromatin condenses into chromosomes. Option B, the proteins attached to each chromatid are cleaved and sister chromatids are separated as daughter chromosomes. Option C, the chromosomes are aligned at the equatorial plate. And option D, the daughter chromosomes are moved and attached to the opposite end of the cell. Now this is a related question, and having listed all the characteristics with the answer in the previous question, this should be an easy one. Which of the following is a characteristic of metaphase? Let me wait for a minute or so and then I'll take a look at the chat box. Meanwhile, keep posting your answers. Characteristic of an animal metaphase. Animal cell undergoing metaphase. Okay, let me take a look at the answers. Which of the following is a characteristic of metaphase? Farsana says option C. Aishwarya is saying option C. Vedashri is saying C. Okay. If you say so, then option C it is. The correct answer is option C. The chromosomes are aligned at the equatorial plane is a characteristic of metaphase. This is the correct answer to the question number 9. And listing the characteristic again, the M phase or the metaphase, M phase called in the cell cycle, metaphase within the mitotic phase, is also the longest phase of mitosis. So this is technically the longest phase of mitosis because The chromosomes have to align at the center an imaginary line, which is also known as the metaphase plate or the equatorial plane, and then each microtubule has to carefully attach to opposite sides or poles of the chromosome, right? Only upon successful attachment of all the spindle fibers to opposite pole of all the aligned chromosomes which are duplicated, only then can a successful cell division happen. So if a cell gets arrested at M phase, it does not proceed to mitosis or normal cell division. Centrosomes are the organelles through from which spindle fibers originate, right? Centrosomes are now at opposite poles of the cell, like this. One is on this side and the other here, and this is the equatorial plane or the metaphase plate that we all were talking about, an imaginary line across which all the chromosomes are arranged and which are joined by now the emanating microtubules from the centrosomes, which are at opposite poles. Chromosomes are located at the metaphase plate. Kinetochores of sister chromatids are now attached to tubules of opposite centrosomes. So this is what I was talking about. Kinetochore is the region of the centromere which is attached to a microtubule, and the microtubules complex is now called the spindle apparatus. So this whole arrangement is now the spindle apparatus with centrosomes at two ends, the microtubule emanating and then holding the chromosome on the opposite poles and aligning on a metaphase single plate. On the right bottom, we see a cell in metaphase, looks somewhat like this. These are the little spindle fibers, and this alignment, this imaginary plate, is the metaphase plate. I hope it's clear to everyone. A short question and an easy answer. Okay, great. Quickly moving on to the 10th question. Lysosomes play a role in dashed. Lysosomes play a role in dashed. Option A, necroptosis. Option B, autophagy. Option C, apoptosis. And option D, necrosis. Now, what do you think lysosomes play a major role in? They are always or most of the times involved in a process. Is it A, necroptosis? Is it B, autophagy? Is it C, apoptosis? Or is it D, necrosis? We have reached the 10th question. We have an hour left or so. The class could maximum go up to 5 p.m. The class timings are 3 p.m. to 5 p.m. We have reached the 10th question. There are 5 more questions. I had prepared 15 questions for you guys. Since these questions are short, and followed by the time calculation for the explanations as well as waiting for you guys to answer, we can easily cover 15 questions within the span of 2 hours from 3 p.m. to 5 p.m. Right? So let us answer this question. Lysosomes are playing a major role in which of these pathways? Okay, there is a comment I see. Can you please say meaning of the options? That is the trick here, right, Farsana? I cannot explain the meaning of these options unless you have answered this. Part of the answer lies in the meaning of these processes. So to answer this question correctly, you have to have knowledge about all these processes. So I will reveal all these processes individually after you have ticked your correct option. I will clear all your doubts. You have to just wait for a minute or so. I'll also wait for a minute to look at some of the answers. Lysosomes play a role in necroptosis, autophagy, apoptosis, necrosis. Let us look at all the answers that I have collected in the chat box. Vedashri says option C. Farsana hasn't answered, but she was asking a doubt. Maybe she's confused. Aishwarya is answering option B, and I don't see any more answers. Did everyone answer? Does anyone more wants to answer this question? You can just take your guesses or your best logical hint or hunch. Don't shy away from participating. We have 5 more questions to go, and then we will end this session. Okay, let's not waste any more time. Let me reveal what is the correct answer to this question. Correct answer to this question is autophagy. Option B is correct. So Aishwarya was correct. Lysosomes play a major role in autophagy. Right? Now let us understand, where does lysosomes play a role in autophagy? Now, what are lysosomes? If you remember, if you recall, lysosomes are special organelles which are filled with acids. So, lysosome compartment is an organelle. Inside the lysosome is a lot of acid and it has low pH. And it is also called the waste manager of the cell. So, any protein or any particle which needs to be degraded by the cell is sent inside the lysosome, where the lysosome can digest the protein with high acidity. So here is a scientific example when there is autophagy induction. And what is autophagy? Auto means self and phagy means eating, self eating. So autophagy is a type of cellular process in which a cell clears its debris or organelles or recycles its proteins, and many other pathways involve autophagy. Now if there is an autophagy induction, then there is formation of a temporary organelle called a phagophore. Now what is a phagophore? Phago means eating and phore means an organelle. Elongation and engulfment of the phagophore and engulfing all these things. So it can be a mitochondria, it can be a virus or a pathogen which is infecting the cell, protein aggregates, it can be degraded products or endoplasmic reticulum, and then forms the autophagosome. So now these autophagosomes are organelles which are temporarily formed when the cell is undergoing autophagy. These autophagosomes now fuse with the lysosomes. So an autophagosome will fuse with the lysosome containing lysosomal enzymes which are highly acidic to form an autolysosome. So this is a fusion between the lysosome and the autophagosome containing all the materials which need to be digested and removed. The virus, the pathogen, the degraded the mitochondria, and this all these components can now undergo lysosomal degradation. The cell can digest any unwanted protein or pathogen or recycle these. So this will recycle all the amino acids present in a protein and the cell can reuse them. So these degradation, proteasomal degradation, lysosomal degradation are machineries which cell use as a waste management system. And whenever there is autophagy induction, there is fusion involvement of lysosome in autophagy. That is why the correct answer to this question was autophagy. Lysosomes are associated heavily with autophagy process inside the cell. Now what are all those other options, as Farsana was asking? Apoptosis is a controlled and silent process that prevents inflammation. So all these are apoptosis, necroptosis and necrosis. I'm sorry. This would be necrosis. Yes. So apoptosis Apoptosis, necrosis, and necroptosis are all forms of cell death. When the cell has to die, or when the cell is dying, it employs any of these pathways. It has several pathways when a living cell can take to end its life or die. Whenever there is cell suicide or type 1 cell death, which is called apoptosis, is self killing or cell death from internal formation of apoptotic bodies like this, which is also known as blebbing. Then there is another type of cell death which is called autophagy. Now autophagy is also one form of the cell death in which can be further divided into macrophagy, microphagy, autophagosome formation, and autophagosome lysosome formation. Then there is necrosis or inflammable, sorry, inflammation and the cell death associated with inflammation and cell damage, and there are other forms of cell death. Apoptosis is a controlled and silent process that prevents inflammation. So the cell will itself kill itself from within, and there is no inflammation. Necrosis is a death mechanism when apoptosis is blocked. So most of the cells prefer apoptosis since it is not causing any inflammation. It is silent suicide killing. But when apoptosis is blocked, then comes necrosis, which can then pour its infected contents, as infected cell can die and pour its content outside, alerting other cells and causing inflammation, often leading to inflammation. Necroptosis represents a hybrid form of both seen in certain diseases. Now, these are some of the salient features of apoptosis, necroptosis, and necroapoptosis. How good it is regulated? Apoptosis is highly regulated. It is caused by two group of proteins called pro-apoptotic and anti-apoptotic proteins. These are all forms of programmed cell death, which are also known as PCD. Necroptosis is regulated, while necroptosis is hybrid. Caspase involvement. Now, caspases are enzymes which are responsible for apoptosis. So these enzymes cleave certain proteins which act as ligands and signal the cell for a programmed cell death. So apoptosis is caspase dependent, while necroptosis is caspase independent, and necroapoptosis is caspases and RIPK kinase dependent. Now, what is the morphology of cell? So there is cell shrinkage and membrane is intact in apoptosis. In necroptosis, there is cell swelling and membrane rupture, while in necroapoptosis, it is mixed. So there is membrane rupture and spillage of the cytoplasm and infected contents in necroptosis, while in apoptosis, there is cell shrinkage, membrane blebbing. Inflammation, it is non-inflammatory. It is inflammatory, the necroptosis is inflammatory, and necroapoptosis is partially inflammatory. And what are the key proteins that help the cell undergo apoptosis? Caspases 3, 7, 8, 9, RIP kinases 1, 3, and MLKL, while in necroptosis we have both caspases and RIPK proteins. So all these are forms of cell death, while autophagy is a type of clearance mechanism of the cell where it recycles its components. Apoptosis is a cell suicide, silent death of the cell, whereas its component is recycled, the infected cell is removed. Necrosis is an inflammatory cell death where the membrane is ruptured, cytoplasm is spilled out, and it causes inflammation, draws attention of other cells, calling and recruitment of white blood cells. And necroptosis is a hybrid form which is a mixture between apoptosis and necroptosis, and it's seen in certain diseases. Is it clear to everyone? So the lysosomes play a major role in autophagy, or the self-clearance mechanism in which cell is planning to recycle most of its component. Okay, great. Moving on to the next question. Question number 11. Which of the following gene is the guardian of genome? It's a nickname given to a following gene or a protein. Which of the following gene is the guardian of genome? Option A, P53. Option B, RB. Option C, INK4. And option D, P10. Now, which of the following do you think is the guardian of the genome? if you can relate to the functions of these proteins, then you can easily answer this question, right? Guardian of the genome. In fact, this is a famous protein. If you just type the name of this protein, you can find hundreds and hundreds of research articles and experiments and its importance in medicinal medicine and translational health, right? It is nicknamed, that is why, due to its heavy impact in medicinal health, and its protective effects, it is nicknamed the guardian of genome. Okay, let me wait for some time and then look at the answers. I'll wait for a minute or so. We still have a couple more questions till then. We will go slow. Understand the concepts behind these answers so that these concepts get engraved in our brain and in our memory. When you encounter these questions or similar questions in your assignments or while you are reading the lectures, this TA session will definitely help you in refreshing or recollecting your memory. Now, which of the following gene is the guardian genome? Let's look at what students have answered. Definitely it's A. Okay. Aishwarya is also saying A. Farsana is also saying A. I mean, it is quite an easy question. And the correct answer is option A, P53, which is also known as the guardian of genome. Well done, guys. You have answered very correctly. P53 is a protein which is involved in many pathways inside a cell. It's a small protein and the name signifies its molecular weight. So its molecular weight is 53 kilodaltons. That is why this protein is named as P53. Protein which weighs 53 kilodaltons. Now, what all processes does this protein involve? It is involved in the cell cycle, in metabolism, stress response when the cell is facing a stress or nutrient deprivation, stem cell differentiation, very important, cell senescence or cell death, aging of the cell, dying of the cell, inflammation, response to infection, programmed cell death, blood vessel formation, DNA repair. P53 is everywhere, you name it, right? You name it, you find it. That is why it is called the guardian of the genome. And what are the other proteins that we saw in the options? What is RB? RB stands for retinoblastoma protein. INK4 is cyclin dependent kinase inhibitor 2A or CDKN2A, is also known as INK4. Inhibitor of cyclin dependent kinase, right? It's a CDK inhibitor. And P10 stands for phosphatase and tensin homolog. Now here is a table citing all the gene functions. RB or retinoblastoma blocks G1S transition by inhibiting E2F. Now this protein plays as a cell cycle checkpoint by blocking G1 to S transition. Now G1 to S, G1 is a growth phase while S is the synthesis phase. If you remember, we encountered a question of cell cycle where I have discussed G1 and S are both part of the interphase, whereas G1 is growth phase 1 where there is cell growth and S phase is a synthesis phase where there is DNA replication. Now it blocks the G1 to S transition. From the cell to move from G1 to S, it has to cross this checkpoint which is retinoblastoma. Now if there is retinoblastoma, it will block the G1 to S transition by inhibiting this factor. And this is associated with many diseases like cancer, where retinoblastoma is mutated. That is why many cells bypass this checkpoint and always keep transitioning from G1 to S, thereby entering cell cycle regularly. So a cell does not know when to stop dividing because there is a mutation in retinoblastoma. Similarly, so retinoblastoma mutation is associated with the disease retinoblastoma, lung and bone cancers. INK4, which is a cyclin dependent kinase inhibitor, inhibits CDK4/6, keeping retinoblastoma active. So this is upstream of retinoblastoma. INK4 inhibits a CDK, thus keeping retinoblastoma in point at the cell cycle checkpoint from G1 to S. Thereby, if INK4 also has mutations, is associated with cancers such as skin cancer or melanoma, pancreatic cancer, and glioblastoma or brain cancer. P10 inhibits PI3K AKT pathway, preventing excessive growth. Now, this is a signaling pathway. PI3K phosphoinositol 3K phosphoinositol 3 kinase and AKT pathway. This is an inhibitor of this, and this is also associated with cancers like prostate cancer, breast cancer, and endometrial cancer, as well as brain cancers. So all these three belongs to, as well as P53, belongs to a class of genes called tumor suppressor genes. These are tumor suppressors. They are rightly named because expression of these proteins suppresses tumors, or a mutation in these proteins is associated with tumor formation and cancer. They work in concert to prevent uncontrolled cell division and tumor growth. Their mutations are frequently seen in cancers, making them key targets for cancer therapy. So, cancer therapy, most of the times, the chemotherapies and drug inhibitors target these proteins like P53, RB, INK4, and P10, so that uh, the uncontrolled cell division can be stopped. Now, is it clear to everyone? You guys have answered very well. This was also an easy question. The guardian of the genome, P53. Good. Great. Moving on to the next question. Question number 12. After which phase of cell cycle do cells enter quiescent phase? Again, a question on cell cycle. After which phase of the cell cycle do cells enter quiescent phase? Option A, G1. Option B, G2. Option C, S. Option D, M. G1 is growth 1, G2 is growth 2, S is synthesis, and M is mitosis. But in the question here is asking about a different phase altogether, which is the quiescent phase. And after which phase of this cell cycle can cells enter the quiescent phase? Can be a bit tricky question, but I hope you can answer it correctly. Let us wait for a minute and collect some answers. I'll check the chat box after a minute or so. After which phase of the cell cycle do cells enter quiescent phase? Okay, let me check all the answers in the chat box. Aishwarya is saying A. Vedashri and Mrs. Babu are also clicking on A. A, G1 is the correct answer to this question. Well done. G1 is the phase of the cell cycle after which the cells can enter a quiescent phase. So G1 phase is an important phase, and the quiescent phase is also known as G0 phase. Right? So here is again the cell cycle with all the explanations and a briefing. So in the cell cycle, let's go phase wise. This is the gap 1 phase or the growth phase where there is cell growth. Then comes two paths. The cell can either go to a resting phase called G0 phase. Cell cycle is stopped, the cell leaves the cell cycle when it is not going to divide, or it can enter the S phase in which daughter cells, so sorry, it can enter the S phase in which the cell duplicates its DNA. Centrosome is also duplicated. In the gap 2 phase or growth 2 phase, cells grow some more and prepare for the mitotic phase, which is the early prophase. So there is nuclear membrane, chromatin, nucleus, doubling of the DNA has already happened, the replication has already happened. Then comes the prophase where the chromatin is condensed, nucleolus disappears. Prometaphase, when the nuclear membrane is completely gone, the kinetochore is attached to the spindle molecules and microtubules, right? Then comes the metaphase or the mitotic M phase, where the chromosomes are aligned on the metaphase plate. Centrosomes on both poles emanate spindle fibers which attach to the kinetochores in each sister chromatid on the opposite poles. Then comes anaphase, where there is pulling of these microtubules towards two poles of the cell. And in the telophase, there is individual nuclear membrane reappearance and formation of two nucleus inside a single cytoplasm, which is followed by cytokinesis or the division of the cytoplasm. And after this, the cell can then enter the G0 phase or the resting phase if it does not want to continue dividing. So G0 is a non-dividing resting phase. Some cells can re-enter the cell cycle, which are known as quiescent cells, or which are poised, which can enter the cell cycle anytime but not right now. While others stay permanently, which are terminally differentiated or senescent cells. So a best example of a quiescent cell is a stem cell. A stem cell is a quiescent cell when it is not dividing, while a terminally differentiated cell example would be neurons. Neurons cannot divide further. It can never it does not generally enter inside the cell cycle phases. And this entry inside G0 is regulated by growth factors, contact inhibition, and tumor suppressors like P53 and RB. So we just encountered these tumor suppressors in the previous question. And G0 phase is important in cancer and tissue regeneration and aging. Right? Is it clear to everyone? You guys have answered really correctly that after G1 phase, the cell can either decide continuing on with the cell cycle or enter the quiescent phase, which is the G0 phase. Cool. Coming on to the 13th question. Which of the following acts as an antigenic determinant in gram-positive bacteria? Which of the following acts as antigenic determinant in gram-positive bacteria? Option A, lipopolysaccharides. Option B, eicosanoic acid. Option C, palmitic acid. And option D, lipoteichoic acid. In a gram-positive bacteria, which of these can act as an antigen or is the primary antigenic determinant? So for clarity, antigen is any substance or any molecule which can elicit an immune response in an individual. That is called an antigen, right? Now on a gram-positive bacteria, which such molecule can act as an antigen inside humans or in any other organisms, which can elicit an immune response? Let me wait for a minute or so and collect some answers. An antigenic determinant in a gram positive bacteria. Okay, let me take a look at the chat box. It's been a minute or so. Vedashri says D, while Farsana is saying A, and I don't see any other answers. Do you guys need some more time to think about it? Aishwarya is also saying A. Okay. It's okay if your answer is not correct. Do not be disheartened. I'll be revealing the correct answer in a minute or so, and then we will understand the rationale behind it so that your concepts are clearer, and in the next attempt you do not make a mistake if your answer is wrong the first time. It's okay, and I will urge all the participants to at least let me know what they think is the correct option. We have two more questions to go. Number 15 will be the last question of this session. Right? So I see some more answers coming up. Mahavir Singh is saying A, Aishwarya A, Farsana A, and Vedashri D. Okay, let me reveal what is the correct answer to this question. Option D is the correct answer. Vedashri has answered correctly. Lipoteichoic acid is the antigenic determinant in gram positive bacteria. Lipoteichoic acid. Okay, so here on the screen is a representation of different cell membranes and cell walls of microbes like the gram negative bacteria, the gram positive bacteria, mycobacteria, and fungus. Right? So let us focus on our question, which was a gram positive bacteria. So this is how its cell membrane and outer surface looks like. This is the bilayer, lipid bilayer cell membrane. There are some lipoproteins, and a lipoteichoic acid is a molecule which is anchored in the plasma membrane, protruding outside towards the external surface of the bacterium. And then the bacterium also has a peptidoglycan layer and some teichoic acid molecules. So lipoteichoic acid is a prime antigen in the gram positive bacteria. And why do these bacteria are called gram positive? Because they stain for gram staining. They are positively staining for gram stain. Gram stain is something which binds to the peptidoglycan layer. Now, since the peptidoglycan layer is on the external surface of the cell membrane, this can readily attach to the gram staining, and thus these bacteria are characterized as gram positive bacteria. Then there are other classes of bacteria called gram negative bacteria, which have a double membrane structure. The cell membrane, inner cell membrane, and there is an outer membrane. In between that is a sandwiched layer of peptidoglycan. So this peptidoglycan is not exposed to the gram staining, thereby these bacteria stain negative for gram staining. Thus the name gram negative bacteria. Now in the gram negative bacteria, what is the thing that is exposed outside? These are the molecules called lipopolysaccharide or LPS. So in a gram negative bacteria, lipopolysaccharide is the antigen, while in gram positive bacteria, it is the lipoteichoic acid which is the antigen. If you compare other microbes, lipoarabinomannan, glycolipids, lipoarabinomannan is another antigen for mycobacteria, as well as mannose proteins in fungi also serve as antigen in humans. So the correct answer is lipoteichoic acid in gram positive bacteria. Now looking closely at lipoteichoic acid, what is lipoteichoic acid? It is a strong antigen that plays a critical role in immune recognition, inflammation, and bacterial pathogenicity. Now lipoteichoic acid is also rendering the additional pathogenicity to these gram positive bacteria. This is the cytoplasmic membrane, and embedded inside the peptidoglycan layer are teichoic acid molecules and lipoteichoic acid, which are also anchored to the cell membrane. And there are other proteins called wall associated proteins. Now, why is this economically important to humans? Because many gram positive bacteria cause disease, and one of the major immunogens or antigen is the lipoteichoic acid. So, targeting LTA or lipoteichoic acid could be useful for developing vaccines and therapies against gram positive infections. Now, these are some of the important features of lipoteichoic acid. Location: it is located on the gram positive bacterial cell wall. What is the structure? It has a glycerol phosphate or ribitol phosphate plus glycolipid anchor. So this is the glycolipid anchor and glycerol phosphate backbone. Immunogenicity: recognized by TLR2 and TLR6 triggers inflammation. So these are toll like receptors present on human cells, especially white blood cells. These receptors recognize these lipoteichoic acid as antigen, and this can trigger inflammation inside human cell or host cell. Immune response is mounted by activating macrophages, dendritic cells, and B cells. This single molecule of lipoteichoic acid can be recognized by macrophages, dendritic cells, and B cells. And it is involved in sepsis, endocarditis, autoimmune diseases. It has a strong clinical significance, and any vaccine against lipoteichoic acid can prove effective against gram positive infections. Is it clear to everyone that lipoteichoic acid is on the outer membrane of the gram positive bacteria, which acts as an antigen, while in the gram negative bacteria it is lipopolysaccharide? Cool. Thanks for the response. Moving on to the next question. Question number 14. Porins in mitochondria allow molecules with molecular weight of dashed. Porins in mitochondria. Option A, less than 20,000 Daltons. Option B, less than 50,000 Daltons. Option C, less than 5,000 Daltons. Option D, greater than 5,000 Daltons. Now, what is the size or molecular weight of proteins which can pass through porins present in the mitochondria? Is it option A, less than 20,000 Daltons? So, protein molecular weight and are measured in Daltons or in kilodaltons. Is it option B, or less than 50,000 Daltons? Option C, 5,000 Daltons, less than 5,000, and option D, greater than 5,000 in porins inside mitochondria. Let me wait for some time and check the chat box for answers in a minute. Okay, let me check the comment box for answer. I see Vedashri is answering option A, and Farsana is answering option D. What about the others? Do others know the correct answer to this question? Or any guesses that porins might be transporting what size of molecules inside the mitochondria, inside and outside? What is the size of the molecule? Well, I don't see any other answers, so let me reveal what is the correct answer. Correct answer to this question is option C, less than 5,000 Daltons. So this is the molecular weight of molecules or proteins which can pass through porins present in the mitochondria. Now, what are porins? Porins are proteins or channel proteins which allow passage of molecules inside or outside of the organelle. Porins are also present in the plasma membrane, which help transport substances like aquaporins, help in transport of water. Porins are also present on the outer and inner membrane of the mitochondria. So this is the structure of mitochondria, where we have uh, different parts. The outer membrane, there are porins in the outer membrane. Then there is intermembrane space. This is the inner membrane, so the intermembrane space, and inside the inner membrane is the matrix. These are also called the cristae, finger-like projections. There is mitochondrial DNA inside the inner membrane, and there are matrix granules as well as ribosomes inside mitochondria. Right? Mitochondrial porins typically allow small molecules and proteins up to molecular weight around 5000 daltons or 5 kilodaltons to pass through, meaning they can only facilitate movement of very small proteins. Now 5 kilodalton is the size of a very small protein. They are not designed to transport large proteins through mitochondrial membrane. That is why they only transport small molecules and not very large proteins, in comparison to the cell membrane porins, which can transport bigger molecules inside and outside of the cell. Right? Moving on to the last question of this session, question number 15. ATP synthase is a dashed shaped multimeric protein complex. Option A, mushroom. Option B, clover leaf. Option C, L shaped. Option D, V shaped. ATP synthase is a dash shaped multimeric protein complex. This should be an easy one for you guys. So I want all of you guys to answer, or whatever you think is the correct answer. Make your guesses. ATP synthase is a dash shaped multimeric protein complex. Option A, mushroom. Clover leaf, option B. Option C is L shaped, and option D is V shaped. If you remember the structure of ATP synthase. I'll wait for some time and then go through the chat box for answers. Any answers yet? I don't see any answers yet. What do you think, people? Do you remember the structure of ATP synthase, or at least the shape of its protein complex? Okay, I think the session has been going on for too long, and it is a time and information overdose for you as well. So let me just move on with this question and reveal what is the correct answer to this question. ATP synthase is a mushroom shaped multimeric protein complex. It was easy answer. It is a mushroom shaped protein complex. So this is how an ATP synthase protein complex looks like, with all its subunits and domains. So this is typically called the F1 F0 ATP synthase. This is from E. coli, bacteria E. coli. The ATP synthase is a large mushroom shaped asymmetric protein complex. The mitochondrial ATP synthase is a multi subunit protein complex having an approximate molecular weight of 550 kilodaltons. If you remember, the porins could only allow 5 kilodaltons. But ATP synthase is one present in the membrane of the mitochondria weighing 550 kilodaltons, together with all the units combined. It has two subunits: F0 unit, the rotational unit, this consisting of alpha, beta, and gamma subunit, which is where the ATP synthesis happens, and the F1 catalytic subunit, which also acts as the proton transport, the proton channel, and the delta and beta subunits. Right? So this is how mushroom shaped ATP synthase looks like. This is the head of the mushroom, this is the base of the mushroom, and it is approximately 550 kilodaltons in size. With this, we have come to the end of this problem solving session. I will just stop presenting my slides like this. Let me come back to, sorry, let me come back to the meeting. Yeah, so far so good. We have covered almost 15 questions belonging to different lecture modules covered in the week 3. In the next week, we will be covering sequentially week 4 assignment questions, exemplar assignment questions. Thank you for joining the class today. The presentation as well as the recorded session, the recording of this session will be uploaded in the YouTube. The presentations will be available in a drive folder which will be shared to you via the NPTEL authorities. If you have any questions, you can ask me, or if you have any suggestions on improving the presentation or improving the class, do you like the class or not, you can post it in the chat box, you can speak up. If not, then I'll quickly end this session. So what I'll do is, for all the people who are attending these classes regularly, what I do is generally I'll paste the YouTube link or the playlist in which I am going to add all these recorded sessions. So I'll do it right away. I'll paste the link in the comment box shortly. Let me just open my YouTube where I can copy the link for you guys. This is a small benefit you have over others who will watch this recorded lecture. Vedashri has raised a hand. Yeah, Vedashri, you can switch on your mic, or if you feel free, you can either talk or post it in the comment box. Meanwhile, I'm just trying to, yeah, this as well to receive the. Okay. Playlist, how to get link. This is the link. Yes. So I'll just paste the link here. So in the chat box, this is the link of all the playlists which has previous week recordings as well as upcoming week's recording will be uploaded in this link. So we are currently at lecture week 3. We will reach till lecture week 12, and all these questions will be from these topics. So please read through your recorded lectures and attend these live sessions. That will be more than enough for you to score good marks in the NPTEL course for basics of biology. It's not a difficult course. It's a very simple course, and there are multiple teaching assistants like me who are trying to help you out, and Professor Trivedi's guided recorded lectures will help you out in understanding biology. If you want to talk to me, you can. Please switch on your mic and talk to me, or I'll end the session soon because 5 p.m. is closing by. Yes.
Good evening, sir.
Yes. Good evening.
Sir, actually, there is the other name. I don't remember the other name for TCA cycle, which is given by the name of the scientist, sir. Can you just say
That is
that?
Krebs cycle. Krebs cycle. It
Okay,
was
sir.
discovered by Krebs.
Okay, sir.
Yes.
Thank you, sir.
You're welcome. Okay, if anyone has no more to say. I'll end this call. Thank you for joining. We'll meet again on next Saturday with practice problem set for week 4. Bye bye and take care. I'll end this call. Thank you.
Thank you, sir.